So a little bit of background. We've touched on this a little bit as well in previous talks, but people would name things for convenience. They would use descriptive names prior to 2007. And studies in high-path avian influenza would use things like Goose Guangdong lineage or Goose Guangdong-like, Qinghai lineage, Fujian lineage. Yeah, okay. So we had these descriptive names, right? We would call classic swine lineage, right? For human influenza. And there was a number of them. And we would often use a geographic location or host as the descriptor, as that a critical part of the name. And that was useful, right? It told us something about where this virus originated from. Maybe it was related to the host, but there are potential consequences and potential stigmatization that comes from that. Well, November 7th, 2006, a paper comes out in PNAS where they described a new variant of avian influenza from China, and it was from a location called Fujian, right? The lineage was named Fujian-like. 2 days later, Margaret Chan, who was being supported by the Chinese government at the time to lead the WHO, was nominated to lead that body, that health body. And then right away, people started saying, well, maybe can this person really be independent, right? There's a paper or an article that came out in Nature on November 15th, right? It was a new variant. People are scared. And of course, nobody wants to have a new virus named after their location. So when that happens, there was a lot of fallout. There was questions about whether viruses were going to be shared amongst different groups or not. And that led to the formation of the WHO, OIE, FAO, H5N1 nomenclature working group. I think this is the first real example of a cross-agency collaboration to address the subclade classifications. Okay, and it unified human health and animal health, both in wild animals and in agricultural animals. The first paper comes out in 2008, has representatives from all over the place. It was led by Gavin Smith and Ruben Donis from the CDC, but they also had members of China CDC that were part of that committee, as well as groups that really span the entire globe. So it was a consensus building exercise, and they came up with a system. Their system was based on if it was a previously named virus, you maintain that name. Okay, so as you're designing new names, right? It was based on phylogenetic information that used all available data at that time. They would use average percent pairwise distance, both between and within clades. I think this is a bit of a problematic criteria, and then they would use bootstrap support based on 1,000 bootstrap replicates and greater than 60% value. So they did put this out to the community. They said, this is the criteria for how you're going to name new viruses. And it was picked up pretty quickly. This paper was describing viruses that spread out of China. There were, I guess this is they're describing the spread from 2003. We had viruses that went into Southeast Asia and to Indonesia, and importantly, they used those names, those subclade classifications. In this group, I think I just realized I need glasses. So in this group, here's the Fujian-like 2.3.4. We have another lineage 2.3.1. We have the Qinghai lineage, the 2.2. These are the viruses that first got into wild birds and then spread across Eurasia, and then some other ones as well. So we were able to classify them, have names, and still use them effectively. This is the current state. We have so many names, all right? And I think a lot of these names are not necessarily useful. Or I shouldn't say they're not useful. They're not necessarily important, right? And partially, I think this has to do with some of the criteria that were used, and this fact that we put out the criteria, or as a community put out the criteria and create this cottage industry of creating new names. But from that tree, we have 2.3.4, 2.3.1, and 2.2 as the main lineages that continue to circulate. We have 2.3.1 has divided into 3 different groups. One that is spread within Southeast Asia, one that is restricted to Indonesia, one that is found in India, Nepal, and Bangladesh. Then we have this wild bird, initially a wild bird lineage that spread throughout Eurasia and then became established in poultry in the Middle East. And that one, when it became established in poultry in the Middle East, this is where it goes from dark blue to light blue over here. It really seems to have diversified from the ancestral population. And then we have the 2.3.4 lineage, which has that global distribution. It is found in wild birds, poultry, mammals, and humans. So the reason why I show this is that pairwise distance will always increase over time as viruses continue to circulate. So that's part of the reason why we start having these new lineages being named. The virus has just continued to circulate, and as it was there, it wasn't like there was a new event. It was continuing to be detected, and it was being given a new name. And I think that this criteria did cause some problems. I think there's better ways, better criteria that have been used since with the classification of SARS-CoV-2. But this particular lineage meant, or this particular criteria allowed for people to fairly easily describe a new lineage, because it was this group of viruses that had just been a little bit more genetically divergent than that ancestral population. And the criteria was 1.5% genetic distance as well, which was very, very small. But we do see that these lineages, because this geographic isolation, so the one lineage I showed 5 over here, I'm not showing Indonesia because it's just in Indonesia. But in poultry in India, Nepal, and Bangladesh, in sorry, that's India, right? Sorry, this is the second one is Southeast Asia. The third one is India, Nepal, and Bangladesh. The fourth one is the Eurasian group. Again, and this is the one that initially started spreading throughout Eurasia, got into Africa, and established in the Middle East. And then the top one, again, that geographic distribution. And this is important criteria in defining these different sublineages. The other thing that's also important with high path avian influenza, or influenza in general, is that this probably has the largest host range of any virus that I know of. I don't know if somebody may challenge me on that, but this H5 will be found in all of these except for the fruit bat and the horse, I believe. But H5 viruses have this massive, massive host range, and the mechanisms of viral spread within these different hosts are different, and I think often underappreciated. We have initially a production system and in Asia that was often had a lot of human exposure. This top picture here is Vietnam, somebody taking chickens to a market on the back of a bicycle. We have Indonesia, a household where there's a chicken in the household. And this is Hong Kong prior to 2003, when they changed the system where people would have exposure to multiple animals. The bottom here are ducks. And then up here are chickens. The people would select a live bird. It would be slaughtered in the back, and then they would take it home. And that was the system that these viruses were transmitting in. Now we're finding it in large scale production systems. This is completely different modes of transmission that is going to drive the diversity of the viruses that we see. So we used a discrete model and tried to actually incorporate this into our phylogenetic analysis. We just added some additional structure. 3 minutes. Okay, we added some additional structure so we can look at how much transmission was happening in domestic birds, to domestic birds, domestic to wild, wild to domestic, and wild to wild over space. Big conclusion: the speed of transmission was driven by domestic trade, but distance was often associated with wild bird. It was linking very, very long distances. 2014 outbreak. In North America, we have 2.3.4 being introduced into North America. Rapid spread across the country. And my slides are missing. That's okay. So it shows up in North America, and it initially gets into wild birds, shows up in the Midwest in February of 2015. As it's showing up in new farms, everybody's saying wild birds. We can't do anything. It's being spread by wild birds. Well, how many wild birds are there in Minnesota in February, right? In particular ducks, not so many. We have evidence of transmission amongst poultry farms, and then the spillover was the early part of the outbreak. One of my former students, Joe Hicks, decided to try and test this. He developed a SIR model that he incorporated into, various models that he incorporated into a phylogenetic analysis. One population SIR, one population with an unsampled source, two populations, chicken and turkey, and two populations, chicken and turkey with an unsampled source. What we saw, we had the two populations, turkeys and chickens, and this was driving the diversity of viruses as they spread. Okay, so we have different modes of transmission that is determining the viral spread. We have it across scale. We have viruses that are being localized in one country in these different systems, and that's actually what's determining the diversity and informing how we name these viruses. The next slide is my final conclusion. Final conclusion is that actually I think we need to actually start incorporating pathogen diversity, not just the phylogenetic information, but understanding that ecology and the epidemiology, because I think these things are what make that naming more broadly useful, right? It's great that we can have so many different buckets, but we might want to know which of those buckets are important. And I think ecology, epidemiology should go into that, determining what of those buckets are important. I think the talk that Betty gave this morning, talking about how you can incorporate those deep mutational scanning and make some predictions, but then also, do we have opportunity to get further input from other groups to figure out which ones are important? Okay. There's some acknowledgements from people that have contributed to a lot of the work and funding. Thank you.
So I'm going to talk about influenza A virus primarily in swine, and it's amazingly simple. There's just three subtypes: H1N1, H1N2, and H3N2. Thanks very much. No, but the thing is that influenza in swine, as we started digging in, and of significant within subtype influenza diversity. So in the USDA, we have a national surveillance system that's been running for about 15 years now. And when we look at that data, we start going from 3 subtypes to 18 different, phenotypically different H1 and H3 HAs. And these are just 2 time series, H1 on the top, H3 on the bottom. The colors represent the different phenotypic clades. So we've got 18 different HA clades over the last 2 years. And we know that the USA is not the world. And we start looking at influenza globally, we start finding a very different picture again as well. And we're fortunate to work within the OFLU network. And in the OFLU network, we're able to ingest data from regions and countries which are participating in swine influenza surveillance. So we've got 15 countries right now. And when you add those 15 countries in, you find another 17 H1 and H3 HA clades. So we're now up to about 30 distinct HA clades across the two subtypes. And the problem here is this is just where we're looking, okay? And historically, and even not just historically now, influenza in swine is generally under surveilled. And I'm going to give you one quick example of that problem and show here a time series from one of our recent OFLU reports. And so you're looking here at the H1 time series for about 6 months of data. And ideally what you're then seeing are all the different colors, which are the HA clades for the H1s. And the orange crosses represent variant infections. So these are swine influenza viruses that have popped up into humans. And these occur in regions in which we have surveillance data, but they're also occurring in regions where we don't. And I'm highlighting one specific case here in Taiwan. This was a human infection acting as a marker for swine influenza in pigs. And there was no genetic surveillance data since 2013 in Taiwan. So humans acting as a proxy for what's going on in the pigs, which is, it's great if you're interested in animal health, though this also represents a zoonotic infection, so not quite as great. So we've got under-surveilled regions as well. So that's another problem. The next problem that we have in our swine data is that the number of groups is not static. I've already mentioned 30 plus HA clades, but there is constant influxes from other animals into our pigs. And I'm pulling up just cherry-picking one example here that was conducted by a postdoc in our group, Alexei Markin, tracking human to swine transmission of the H1 pandemic virus. And what you're looking at here is a timescale tree of about 15,000 HA genes. The black branches represent human HA gene sequences. The orange represents swine HA gene sequences. And we can track the number of human to swine transmissions across this phylogeny. And there were nearly 400 of them in a 10-year period. A subset of those persist. And that is a universal problem for H1s and H3s. Human to swine transmission, and so the emergence of new lineages, entirely new lineages from humans and other animals. And then once those viruses are in our pigs, they have the next problem. We don't have recombination, fortunately. It's a segmented genome, so we have reassortment, which makes it a little bit nicer. But once they get in there, they start to reassort, and that really changes the evolutionary landscape. So what you're looking at here are two NA phylogenies, so neuraminidase phylogenies, where we're mapping HA reassortment events, and those are indicated by the black arrows. So in this specific example, there are 9 major reassortment events. So the neuraminidase gene acquired a new hemagglutinin. And when that happened, there was epistatic changes across the HA and the NA. And that dramatically shaped the evolutionary rates of those different lineages as well. So the pre-reassortment evolutionary rate is the lighter blue, shallow line. The post-reassortment rate is the steeper line. And what was going on here is that following reassortment, there was this period of epistatic change in both genes, and then the virus started evolving faster. So you get more rapid antigenic drift as well. And because of all of those problems, essentially a reassortment, novel introductions, and a wall of diversity, a team of experts in the OFLU network, so this is a tripartite organization, got together to try and develop a system to track this diversity over time. And the basic system that was developed is very similar to the avian influenza system that Justin just introduced. And that followed this first protocol. Everyone's using names out there. So the introduction was, we want to try and maintain those existing names, but remove the stigmatizing or incriminating toponyms. So make it simple and objective. We wanted to follow the evolutionary history of the virus because we know that there's a tight integration between human influenza and swine influenza and avian influenza and swine influenza. So we want to maintain that evolutionary history. We wanted statistical support and genetic diversity accounted for, and we wanted to make it epidemiologically relevant. So we wanted it to matter. We didn't really want to worry too much about viruses that had been detected 50 years ago. We wanted to know what was going on right now. And so we imposed rules that expected clades to be erected if there was continued circulation of the viruses, and there had to be a minimum number of contemporary genes in establishing those names. We did have exceptions, though. because there are always exceptions to these types of rules. And that was that we wanted to incorporate antigenic or phenotypic information or serosurveillance information, and then also reflect public health requirements. So reflect human variant detections. And with these systems, we're able to establish a naming criteria, much like this on the screen here. So this is the H1 phylogeny. And from bottom to top, you're seeing the different general clades and lineages. So we have the 1A lineage, which is reflecting the 1918. I'm just introducing a new pandemic. Everyone's ignored that one. Just flew under the radar. But the 1918 pandemic circulated in pigs, and then this was what seeded the 2009 pandemic as well. So the 1A lineage. Then we have the 1B lineage, which reflects multiple human to swine transmissions of pre-2009 H1 seasonal viruses. And we have the 1C lineage, and that reflects a Eurasian avian lineage that was a wild bird spillover into pigs. So we have this host information. So using this skeleton, this framework, we can track back the evolutionary history of these viruses. With the H3s, it's a similar type of dynamic in that we can look at all of the H3 viruses in pigs, and they're all derived from a human to swine spillover. And those spillovers happen conveniently by decade. I'm telling you it's convenient because we imposed that framework. But we can name these viruses based on the decade or the time at which they were introduced into our pigs. And then we can track the evolution through time following that framework then. Okay, so the really fun thing is the next question, which I think a lot of the discussion is around. Once we have our framework, how do we know when to make new or refine old names? And when we were putting this framework together, that was a critical idea for us because we knew that the viruses were evolving, we knew we had under-surveilled regions, and we knew that we were going to have to respond to public health outbreaks and variant detections. And so the first case I'm going to give you is that public health case, responding to human variants with naming new clades. And so we have a Eurasian avian lineage, the 1C H1s. And the little box that I'm highlighting right there was a clade of viruses where an investigator had gone and sequenced influenza in pigs in Spain. And they'd turned up a handful of viruses. And that was all in one location at one time point in 2019. And so we knew it was there, but there was no evidence of persistent circulation. So there was no point in appending a name to that because it wasn't epidemiologically relevant for us. But it was in there in our backbone and our phylogenetic backbone. And then all of a sudden, about a year ago, there was a human variant detection, 2 years ago. It would be convenient if we're in 2022. I'm behind. Anyway, in 2022, there was a human variant case. So there were 3 years of undetected circulation in Spanish pigs. So clearly that virus is out there somewhere in the pigs. We just don't have the data. So we then had to quickly establish a new clade name for public health. So that was the establishment of a new clade that just fit on our scaffold. So we have this backbone and we can just add a new number. And on we go. The next case was splitting existing influenza case to reflect phenotypic diversity. And this is an enormous wall of data. This is probably around about 1,000 HA genes here. It's from a major clade that we get detected in the US, and also it's been detected in South Korea. There's statistical support, but it's incredibly low diversity. So all of these viruses, all about 2.5% nucleotide divergence from each other. So high identity. But there's a lot of viruses. We're getting 300 to 400 detections of this gene every single year. So what do? We used hemagglutination inhibition testing, and the viruses, representative viruses from each of these three clades were phenotypically distinct. So there was a 4-fold or greater loss in cross-reactivity in those HI assays between the clades. So the three viruses, the three clades are phenotypically distinct. And beyond that, there were human variant detections in clade 1 and clade 3. So then we didn't have the genetic evidence to split, but we had phenotypic evidence, and we also had variant detections as well. So we then appended new names into 3 different groups. The last one is where we don't have human data, and we don't have human variant detections, and we don't have phenotypic information. Can we just use the genomic information to split? Now, I always view genetic information and diversity as blobs or big blobs of diversity that are out there. And then we're sort of trying to constrain all of that diversity into little boxes. Thank you for 3 minutes. Talk fast, yes. I think I've got 5 slides, so I can slow down just a tiny bit onto the really fun part of this. How do you constrain the diversity objectively into these little circles, these little colors, the spectrum? What's all out there? How can we divide it? And so Alexi Mark and a postdoc in our group here developed a really nice objective function. So that means we've got a goal, and then we can maximize and we can also minimize given that goal, which is a really nice way of thinking about this. So how can we maximize coverage? How can we minimize coverage? Those extents. And this algorithm has 2 major sampling strategies. So given all of the diversity that's out there, we can say, choose the 3 best representatives. And then the algorithm will find objectively the 3 best representatives that are out there. We can also impose a threshold, a radius, and we can say, we want to cover all of the diversity that's out there, given a little bit of wiggle room. So if we want to cover 99.5% of all the diversity, how many representatives do we need to choose? And then we can merge those 2 together. How much diversity can I cover if I can only pick 10 viruses or 10 HA genes or 50 genes or 100 genes? How much diversity can we cover? So you can put bounds on what you've got out there. So these circles, try and imagine those circles throughout everything else that I'm going to do. Now, the really nice thing about this is that you can define prior groupings on phylogenies, and you can rescale your trees given different weights. So in this case, we have an established clade. And if we say our threshold is 3% divergence, then the algorithm will automatically spit out the viruses that are not within that circle of diversity. And in this example, this is just a toy example, sorry. In this toy example, there are two groups of viruses that are not within 3% of everything else that has previously been out there. So then you can dig into those 2 groups and say, are these epidemiologically relevant? Do we want to revise and update our naming? The next type of example I'll give you is that you can include other metadata. Thank you. And you can rescale your tree using that metadata. So this is now a tree of human H3 HA genes rescaled using hemagglutination inhibition assay data. So we're using a sort of a 2 antigenic unit threshold, saying that everything within 2 antigenic units is phenotypically similar. You rescale it, and then what you get of the 2,000 HA genes, it all gets squished into a flat line, right? Because they're the same. But there is one area of this tree in gray in the center that is phenotypically novel. So then you can look into that area and say, hey, this is a new group of viruses that we need to look into, potentially name, because it is significantly phenotypically different from everything else out there. And that metadata can be rescaled using almost anything you can think of. This is hemagglutination inhibition data, but you can use other stuff too. Okay, so the really cool thing that we've been able to do is work with the BV-BRC team and implement all of these naming criteria as well. And so Christian is here in the front row, and so I'm not going to go over this, but we're able to distill all of our diversity for the H1s and H3s using this algorithm again. So we can take the 15,000 HA genes and we can distill that down to a topological backbone. And then we can pass that across to the BV-BRC team, and they're able to do classification of unknowns and also database annotations. So this is the user-friendly implementation of the naming scheme. People can dump their data in and they can get an answer. And this is an iterative process because every 6 months, we do the OFLU reports, which is integrated within the influenza discussions at the WHO. So every 6 months, there's a revision. Is there new diversity out there? Do we need new names? Can we update it and then pass it on to the BV-BRC, and hopefully also with Richard onto NextClade too. So we have user-friendly tools everywhere. So to wrap it all up, swine influenza is challenged by many of the similar problems that people in animal viruses have: under surveillance, human to swine transmission, and evolution. We've developed a system that relies upon the evolutionary history of the viruses, and we have a requirement for epidemiological relevance. We have a tool, and you can find our software at our GitHub page. And thank you very much for your time and the funding for everyone. Thanks. Have a good day.
So first, thanks to Elliot for inviting me here. I'm a little bit embarrassed because the entire morning was very sophisticated, and I'm going to get very primitive. So there are a lot of virus groups out there for which any kind of very detailed sublineage classification has not been necessary. It's interesting for me. We could theoretically use these little families to prospectively develop systems so that when it's actually important, we have something ready that makes sense. But because there is no need for it, nobody wants to develop them. And so then we're going to end up in the SARS-CoV-2 situation where we don't know what to do with all the sequences. So that's why I'm talking about filoviruses today. Also, like I have been playing with the ICTV for a very long time, and the only thing that means is I know really nothing except how to be in a lot of fights. And so ironically, most of the fights are not about classification, which by now I consider simple, and 99% about nomenclature, which is really the tough problem. So for disclaimers, I speak for myself, not for the ICTV, not for my study groups, not for the NIH, not for all the other people. And on top, there's a quote from, well, I have it from Charlie Kellisher. I don't know what actually the root is, but that refers to all those fights. Okay, so the first thing I want to do is I also want to change the title of my slide. For one reason that we can either discuss later in a coffee break or tonight at dinner or something, but for philosophical reasons, because I feel pretty strongly that the word classification is wrong. So in all taxonomies, you classify something down to the lowest level of a taxon, which is species, and then the thing is classified, and everything else that you do below it is grouping. And so therefore I change it to subgrouping and group nomenclature. And I will have a fist fight with Peter over this later. So. Okay, so the Q&A questions were, how a virus is classified and what is necessary for the classification. And because the Filoviridae are so small, I can actually talk you through the entire classification system of the Filoviridae, including the subspecies stuff. So how are they classified? Well, all the RNA viruses that encode an RDRP or a reverse transcriptase are in realm Riboviria. They are grouped by the RDRP palm domain phylogeny into different groups. So that's really the first step. And the nice thing so you see on the left, all the RNA viruses that do this, and on the right, you see a blow up of branch 5, and then you see with the arrows where the Filoviridae are located. And the nice thing about Filoviridae, it's such a nice little group. It causes no problems. It's a very, very concise group. And so that is ultimately the classification. And of course, what happens when you classify things just based on an RDRP sequence or like a little bit more, inevitably somebody is yelling about you have no phenotypic characteristics and da. So you try to ascribe certain properties to all of these individual taxa, and you can actually do this very nicely with the Filoviridae. You can start on the top with the Riboviria. I said it's either an RDRP or an RT. Then you go down one level to orthonairoviria. These are only the RDRP viruses. Then Neganoviricota. These are all the negative sense RNA viruses. Then the Hapleviricotina, these are not cap-snatching viruses, and you go all the way down. And so by now we already have a lot of properties of what Filoviridae are, and you have a concise group of Filoviridae based on the RDRP sequence. Now the viruses are very simply organized. They're non-segmented. They do us the favor of having a core set of genes, 5, all of them that are also always in the same order. So if you find something that has those five open reading frames and they're cognate to the other ones, and you have an RDRP and whatever, you can be pretty sure it's a filovirus. So that's very straightforward. And so here's an overview of how the family currently looks like. So there are 9 genera with a bunch of species. You can see the trees. Also, this is a coding complete tree. So that answers one of the questions in the Q&A. Coding complete sequences are necessary and required by the study group to classify these viruses. You can see the bootstrap support values are nice. So again, all very straightforward. So how do we classify then the individual viruses into individual species? We're using PASC, which is just a pairwise comparison sequencing tool that the NCBI developed a long time ago and has not really updated, but I hear from the creator that there's a new system coming called Vista. And this is very nice because you get very nice peaks, very nice valleys, and you can assign demarcation criteria very nicely, and then you can create all your species. So it's all wonderful. So we, and what I mean with we is typically the study group and then a bunch of people who discovered novel viruses thought we should bring some chaos into GenBank. And so that is really, has nothing to do really with phylogeny at that point. It was really just everybody named something differently. And we wanted to come up with some kind of a nomenclature that is logical. And so the first thing that we wanted to do is decide which ones are the viruses that we really, and I'm going to get in hot water when I use the word with the ICTV, but what are the type viruses for Filoviridae? And so because there were so few viruses, it was quite straightforward. We picked the one virus of each species that was the best characterized, which is the one that had the most papers. And we just said, okay, this is going to be it. And then we compared all other novel sequences against this particular virus to build things. And so we made decisions. These became then the RefSeq entries for the individual species. And then we could create a very nice algorithm. So this is outdated. It only has 3 genera in there. But basically an algorithm that says, here's your sequence, you do your PASC analysis, you get your value, you put this value in, and if the value is higher than this, you're over here, and if this value is lower this, and then you're over there. You just walk through all the algorithm, and in the end you say, oh, this is a novel Ebola virus, or this is a novel genus, and so on. So, and all of these activities come from a very long time ago. So they're all 2013, 2014, 15, and they haven't changed because the community has seen no need to change them so far. I said these viruses are easy in quotation marks. So this is a slide, you don't need to look at the details, but these are all the filovirus disease outbreaks in history. So you see there's not many. There are overall some 35,000 cases, and pretty much 30,000 go back to a single outbreak, and all the other ones are very small. And there are complications that make the classification of isolates and strains very easy. The first complication is we don't know the host. So we can't do the nice little chicken turkey game. We only know these sequences from humans. There are only 2 genera that have viruses that cause disease in humans. All the other taxa in this family are represented by 2 or 3 sequences. So we don't need to think about anything there. And there's always, for all the outbreaks, with the exception of a few recrudescent disease cases, all the outbreaks start with a single introduction from this unknown host into the human population. And what that means is that if you have a large outbreak like the Western African one in 2013 to 2016, where you had some 20,000 cases, that all these 20,000 cases can be traced back to that very first single introduction, which means you automatically have a wonderful monophyletic clade, which means, of course, that all these viruses are more similar to each other than to all the other outbreak clades. And that means you can give that a name. And that's basically what the filovirus community wanted. They just said, like, well, every outbreak gets one virus, and that one virus gets one name. We call that a variant, and then it's represented by, you know, tons of isolates and stuff. But who cares? Because if you have Ebola, and you have a tough life. So we don't have massive therapeutics available, but there's a few antibody therapeutics that are in clinical trials and so on, and a few that are in current best practice for the hospitals. But so we're looking a little bit at mutation escape. But overall, Ebola is Ebola, and the clinician only cares about this. Now, where the variant names become important is for tracking them forensically. So this is something that I haven't heard so far here in terms of stakeholders, because these are select agents. These are things that need to be locked up. They need to be tracked. The FBI wants to know where they are. So that's one thing. And then the other thing is, like, there are very few BSL-4 laboratories that can work with these viruses. So it becomes very important for the people who work with the viruses, which virus they're going to work on, because you cannot do 16,000 experiments or repeat and say, like, oh, this was nice, but let's try a different strain. It's very expensive, very difficult. So there was a huge fight for a while of whether we should use the old 1976 best characterized Yambuku variant or then the new Western African Makona variant. And that's why the names were important. I also want to point out that these names were chosen with the African stakeholders, and there were 0 problems about toponyms. And in fact, it was typically the African scientists who wanted toponyms and not us. So I would be a little bit careful with just generally assuming that these names are universally not good. And of course, these names have advantages because you can talk about them very easily. We heard this a little bit in the morning, that communication and presentation is important. And I rather say Makona than 2.7, 3.B. And ideally, of course, you have parallel systems where you can cross-connect. So what we wanted to do is create some kind of an isolate name or variant name that we can put into GenBank in some way, that can be parsed computationally in some form, and that can also be used in writing or in presentations. And so we got heavily influenced, of course, by the influenza nomenclature, and then also by the rotavirus nomenclature, which I've assumed I'll be here after this talk. And so we came up with this ultimate scheme. So on the top, you see the string of information that should be present. And all of this is based on the idea that people want to see the metadata in a publication and not go first to GenBank and then download something. And so this is fairly short. And for all the fields individually, we created rules. So the first field is the virus name, but that was relatively easy. Then we have a strain field, which is typically not filled. And so actually the filovirus community and a bunch of other communities define the difference between strains and variants and isolates and so on. And then we just filled in, like, species, and whether, for example, here the TC stands for tissue culture adapted, or whether it's wild type, that would be a WT and so on. And then we created abbreviations for all of these things. And so now we can, of course, write an easy algorithm and pull all of those viruses out of GenBank and say, show me all the Liberian ones that were tissue culture and not wild type, and everything is right there. And then we applied this to novel outbreaks. And then we applied this to GenBank entries, and it's pretty well adopted by the community, where we use the isolate field to put that string of letters in. And then this is how this looks in terms of the full name, a shortened name, an abbreviated name, and so on. So it's quite straightforward. And then we created the same system for laboratory adapted viruses. So for those viruses that, for example, infect animals that it doesn't infect in the wild, guinea pigs and so on. Typically, you need to serially adapt these viruses and modify them. And so therefore, we use the same strain, but we created new rules. So for example, in the strain name now comes the institute abbreviation where something was created. And then we created a table for all the institutes, simple because it's BSL-4, there are not that many in the world that should be used for these things. And then we did the same thing for recombinant reverse genetic systems, artificial viruses. Again, the same string of fields, but with different information in it. So here you have this little suffix, the REC stands for recombinant. And then you put in the starting virus. In this case, it would be a Kickman variant. And then you name your individual plasmid ABC1. And if you derive from that a second virus, this could be ABC2, and can theoretically create a list of those. So is the current subspecies classification for Filovirus sufficient right now? Absolutely. There's no need to do anything new. Is it in the future? Well, it will be a catastrophe the moment somebody discovers the host. And then we don't know what to do. And so it would be very nice if we could come to a conclusion how to deal with these families and have something ready for that point in time. And then the other question is more specific. Are there specific mutations or common patterns for genetic variation that impact pathogenesis? And for Ebola virus, we really don't know. There are certain mutations that become fixed in outbreaks, especially in the bigger ones where we can trace over several generations. But what they actually do is unclear, and the field is they don't know what to do with it. So that's pretty much it. Acknowledgements, well, all the Filovirus study group members and all the ICTV people who bear with me and so on.
Okay, so thank you. So as you may or may not be aware, there are 9 species of rotavirus recognized by ICTV, but really the majority of rotavirus that we see belong to rotavirus A, B, and C, which have a huge diversity of mammalian and avian species that they infect. The rest of the species typically only infect avian species, or occasionally small mammals like shrews and grouse and things like that. Rotavirus is kind of interesting because we classify not based on RDRP, but based on the VP6 gene. And that kind of creates some kind of issues when we try to compare to other rotavirus classification systems that do work on RDRP. So rotavirus is a segmented genome. It has 11 segments of double-stranded RNA. It's got a relatively small genome size, but it does evolve at quite a fast rate. Recombination is quite rare in lineages that are thought to have been derived by recombination events and then sustained transmission. But reassortment really is the driving force of diversity in rotavirus. We don't see reassortment events occurring between different rotavirus species. So a rotavirus A and a rotavirus B don't reassort. But within rotavirus A, there's an immense amount of reassortment that creates viable progeny viruses. And when you then consider the amount of zoonotic transmission that can occur and the amount of subsequent reassortment events, the diversity is immense. As mentioned, we do have a nomenclature for rotavirus strains, which is the rotavirus group A, B, or C. The species of origin, which is really important for rotavirus because of the huge diversity of species that rotavirus can infect. We also have the subtype of whether it's wild type, tissue culture adapted, a vaccine strain, the country, the isolate name, the date of collection, and the G and P genotype. So we only have classification systems for group A, B, and C. I'm just going to talk briefly on group B and C because there's very few sequences that are available for these viruses. The group B classification system was proposed in 2018, and basically all the sequences that were available in GenBank at the time were analysed, and those with greater than 80% of the open reading frame were included in phylogenetic analysis, and based on the topology of the trees, with strong bootstrap support, these and nucleotide cutoff values, different genotypes were designated. This system hasn't been updated since it was proposed in 2018. There have been no updated genotypes recognised, and very few sequences have been updated in GenBank as well. We don't have an online classification system for group B rotavirus. It's simply based on recreating those phylogenetic trees to propose new genotypes or to classify strains that you might be sequencing. Similarly, for group C, there's a relatively small number of sequences in GenBank, and a whole genome classification system was proposed in 2017 following the exact same method of 80% open reading frame for each gene and strong bootstrap support within a phylogenetic tree. There's been no updates again for group C rotavirus in terms of the genotypes that have been recognised, but there have been quite a number of sequences that have been submitted to GenBank that are untyped. Group C rotavirus doesn't cause a lot of infection in humans, but it's really important in the pig industry. And so it would be beneficial to have a classification system for group B rotavirus or group C rotavirus that is being updated because it's an underrepresented source of mortality in piglets. And knowing the diversity of strains that we see globally would help inform veterinary practices. And again, there's no online classification tool. But group A rotavirus is the most well-characterized rotavirus species. It infects almost every species you can think of. Humans, mammalian animals, avian species, aquatic animals, across the board, where you look, you will find group A rotavirus. Historically, we use monoclonal antibodies to classify rotavirus based on the VP6 protein, so the inner capsid protein. And then in 1989, they moved to, similar to influenza, on the two outer capsid proteins, the glycoprotein VP7 and the protease-sensitive protein VP4, to designate G and P serotypes. This existed up until 2008 when a hemi-nested multiplex RT-PCR system was adopted, where you read the amplicon size on a gel and that dictates what your G and P types are. The reason I bring this up is that the global rotavirus surveillance network run by the WHO and many national surveillance programs still use G and P genotyping today. Sequencing is not that routine for rotavirus globally. We only sequence a very, very small proportion of the cases that we see. And sequencing is not routinely done by WHO. So we always have to come back to this existing G and P genotyping system when we think about the sequencing-based classification that we use. And there are some RT-qPCR assays for genotyping, but they're not routinely used. So the rotavirus classification working group was established in 2008 by Jelle Matthijsens from KU Leuven in Belgium. And this came about because as full genome sequencing was increasing, we knew we had a problem with how do we describe the immense diversity within rotavirus. And so the group B and C systems really came from the group A system. So we had 26 members around the world who came together to talk about how we were going to do the rotavirus classification. And we came up with the nucleotide similarity cutoffs based on the phylogenetic trees with strong bootstrap support. It's not perfect, but it's what we had at the time. And as new sequences are generated, when we think that they don't fall within one of the recognized genotypes, they then get manually classified by Yele, and then he sends out a report to all the working group members, and then everyone votes on whether or not we think this is a new genotype or not. Again, not a perfect system, but it's what we have. And you can see by the number of genotypes within each of the 11 genes, the sheer amount of diversity that we already have well characterized within rotavirus. And this is just at the genotype level. We did have an online database for rotavirus classification called RotaC, but due to lack of funding, that stopped 3 years ago, and we don't have anything that is directly reflecting what the classification working group is designating. And a lot of the new genotypes that we designate as the working group aren't then being uploaded to GenBank by the researchers who are generating the sequences because they don't care. They've done a metagenomic analysis of, a shrew or a bat or whatever and found this rotavirus strain, and they don't care. It wasn't what they were looking for. So last year, I went back and contacted every group who had a new genotype that wasn't in GenBank, and I did the GenBank submissions for them because I wanted that data. Like, you're the only people that will actually care that I did that. Because it matters. If you're going to blast something, you don't know that it's already been sequenced or something similar exists. It just complicates life. So anyway, that was my service to the field. But there are 2 classification systems that are available, but they're not up to date. There's a group in the Netherlands that hosts a Rota-A classification system. It's purely based on the phylogenetic tree that they generate. It hasn't been updated since around 2015 in terms of the known genotypes, and it also incorrectly assigns some of the newer genotypes as old genotypes because it doesn't know that the new genotypes exist. So that's a bit of a problem. And then BV-BRC has a tool. Again, it doesn't seem to be quite up to date in terms of the number of genotypes that we know for each gene. But I haven't found any errors in what it calls as the known genotypes. It's just the newer genotypes. It just comes up with a box saying, we think that this is a new genotype, contact the rotavirus classification working group. But in actual fact, that strain has been genotyped. It's just not updated in the tool. And speaking to Yele, neither of these tools are contacting him around information for the new genotypes that get released every couple of months. So I think that's definitely something that we can work on coming out of this meeting, is making sure everyone's aware of how the updates get released, because it's not obvious. If you don't work in the field, you would never know that this is happening. Sort of briefly touch on semi-curated databases because they're really important for rotavirus. We have virus variation. And when you look at virus variation, it doesn't have all of the sequences that are in GenBank. It only has about half of them, and I'm not really sure why that's the case. And also, you have all these classifications where you can only have to just pull out all of the VP7 genes that are a certain genotype. It doesn't do that accurately. And again, I don't know why, but this creates huge problems when people try to make phylogenetic trees for papers, purely using a database that they think is up to date, but it's not. I know virus variation is going to stop existing and NCBI virus will take over soon, which is up to date, but you can't filter by genotype. So pros and cons, I guess. There are some emerging classification tools that are being developed. And this is in the pre-reading that I suggested. These are really, really great efforts to drive the field forward. What they did with this in particular is they just went, all of the really, really divergent it only or in divergent new genotypes where there's only less than 10 sequences, we're just not going to include those in our algorithm because we can't train the algorithm on such a small data set. I understand why that happens and why that's a complication, but you're then just ignoring a whole bunch of diversity that exists. And then if people have strains that have those genotypes, they're never going to be able to be used with that tool. It also didn't fully align with the old classification working group GNP typing scheme. And models won't necessarily 100% correlate, but when you have a tool that is existed for such a long period of time and has been well adopted, a new tool does need to bridge that gap. It only within the field have made some attempts at lineage classification systems for individual GNP types, but it's not consistent. We've got a semi attempt at a classification system for non-capsid genes for genotype 2 viruses. This hasn't been updated in years. It's not a tool that's been developed with any particular algorithm. It's just a tree has been generated, and based on the phylogeny, lineages or clusters have been designated. This doesn't hold up over time, and when you resolve your tree using Raxamel or IQ tree versus a tree that was made in Mega, your lineages don't hold up, and it becomes quite messy to try and refer back to this system. So we are in desperate need as a field for a better lineage classification system. Another issue with rotavirus is we go and sequence old isolates all the time, like isolates that are 20 or 30 years old and that haven't been previously sequenced. And when you then put them into a tree with existing lineages, it can create a lot of problems. I guess there are a lot of issues with rotavirus classification. With the immense diversity that we see, but also, sequencing isn't routine for rotavirus and it's often delayed. We often sequence strains, 3 to 5 years after they were characterized. Because there's no money within the rotavirus field. This is now an infection that predominantly affects low and middle income countries. They just don't have the resources to be doing routine sequencing. Strains that are emerging via zoonotic transmission can drastically alter tree topology and can make classification problematic. It only do need automated, more up-to-date genotyping tools, and the existing tools that we do have need to be brought forward and incorporate some of the newer technologies and newer methods that have come out of the SARS-CoV-2 pandemic.
So I'm going to talk about the virus that looks like this here. And I'm not mentioning the name quite yet because I'm going to first talk about nomenclature, something that is certainly near to all of our hearts, perhaps not necessarily dear to our hearts. So monkeypox virus, the virus has had a lot of publicity lately, or certainly in 2022, when the outbreak began, about the nomenclature that's used. So I want to use this as an opportunity to remind everybody about the different names that are relevant when you're talking about a virus. So you have the disease name. Originally, that disease was called monkeypox virus. We now call it Mpox, and the disease name has an official body, the World Health Organization, that determines what that name is through their ICD-10 and ICD-11 codes. So Mpox now for what we used to call monkeypox, COVID-19 was determined in that same way. The virus itself, generally that name comes from whoever discovered it or whoever published about it first. And so the virus name can be pretty much anything, and there could be multiple virus names for the same thing that people are talking about. So it's created by the discoverer. If it gets updated, it's a scientific community in one way or another that has to change usage. Clades, as we know, we now have clades that I'll talk about for monkeypox virus. We have our well-defined clade names for SARS-CoV-2. Those, while they don't have an official body that determines what the clade name is, generally go back to some unofficial grouping of scientists who have the particular interest in that virus and the clades of that virus to determine the nomenclature that's going to be used. We've heard multiple times about multiple different viruses today, about different clade names. And then finally, species names, of course, and other taxon names are the responsibility of the ICTV. Currently, monkeypox virus, the virus has a species name that's the same, monkeypox virus. That will change in the next release to conform to the binomial nomenclature. It'll become orthopox virus, monkeypox virus. Severe acute respiratory syndrome related coronavirus, that species will become beta coronavirus pandemicum in about 2 weeks with the official next ICTV release. So I will talk about Mpox, the disease, most of my way through this, but I am talking about the virus. And so I still use that name, monkeypox virus, to refer specifically to the virus. Okay. So human orthopox virus disease, of course, the prototypic orthopox virus disease is smallpox caused by variola virus. That's been known, or a disease that most probably was smallpox, goes back some 3,000 plus years ago. Mpox is pretty much only known since 1958, when it was first discovered in a colony of monkeys. And in those initial years, it only popped up in colonies of monkeys that had been imported to various regions of the world. The first human case was only in 1970. And the reason for that is more than likely that the world population was immune to monkeypox virus before then because of the worldwide vaccination campaign against smallpox. But once vaccination stopped, the world slowly has become more and more monkeypox can be now transmitted to a greater extent because that immunity has waned over the years. Smallpox, of course, can have a fatality rate upwards of 30%, whereas generally Mpox only causes a fatality rate of 0 in many outbreaks, but depending on the clade you're talking about, upwards of 10%. And there are distinct properties of distinct clades, both in smallpox and in Mpox, that can determine differing aspects of its pathogenicity. Sorry. Okay. But up until around from 1970 to 1986, Mpox was confined to Africa. And we came to see that 2 distinct clades existed based upon their phenotypic properties, the disease properties, and they tended to segregate according to different regions in Africa, a region from West Africa, a region in the Congo Basin, and we now refer to the Congo Basin viruses as a distinct clade. That is, they do have a separate monophyletic lineage from the viruses in the Congo Basin versus what we now call clade 2 in the viruses from West Africa. There was the first outbreak outside of that region in 2003 in the Midwest US due to the importation of rodents to be pets into the US. Those rodents, when housed with domestic prairie dogs, then were able to transmit the virus from the prairie dog to the humans. There was no human-to-human transmission. It seemed to all be zoonotic transmission. And in fact, that's a defining feature of that 2003 virus, which we now know came from that clade 2 West African virus. And that is, human transmission is either negligible or very limited when you look at those particular viruses. Now, we look now more recently at 2022, the outbreak that occurred that summer, and you can see here it peaked around about August or so, and then started declining, but it's still with us. So overall, we had 95,000 cases. The mortality rate is about 0.2%, which is consistent, I guess, in general with that clade 2 virus. And we now know that is definitely a clade 2 virus. And it's still with us today. So there were still, 700 cases come this past February, again, with that 0.2, 0.3% mortality rate. Okay, and so how do we distinguish between subspecies variation versus species variation. So this goes back now to what we do in the ICTV to determine whether or not something belongs in a distinct species or genera or subfamily for something else. We define demarcation criteria. So the demarcation criteria tell us whether or not this newly isolated virus is similar enough to an existing virus to belong in that same species, or if it's distinct enough, it may require creation of a new species, new genera, or a new higher level rank. And those criteria initially were all phenotypic based because the ICTV started in the 60s. There were no sequences back in the 60s. So ICTV did its classification solely based upon phenotypic characteristics. As sequencing became available, as we've certainly heard today, sequencing became more and more the definitive demarcation criteria that were used at the various ranks, certainly ranks from species on up to family and order. And so today, for pox viruses at least, you can see that nucleotide divergence is an important criteria, though the numbers get really compressed. And so for different species, we're talking about in the 95, 96% divergence rate to determine whether or not something belongs in a new species. Isolates can be greater than 98% identical for these large double-stranded DNA viruses. And so mostly we use phylogenetic analysis to make the final determination of when something is a distinct species or would be a member of an existing species, and therefore your divergence represents subspecies level demarcation. So this is the tree, the whole family tree. The orthopox viruses are up here, fairly compressed, along with the two clades of monkeypox virus in this case. This is the orthopox virus tree, and you begin to get a feel for what demarcates one species to another. Each one of these here is a separate species. And then when you look with the two clades of monkeypox virus, you see that the branch lengths are a good bit shorter, as they are for clades of variola viruses, in this case, variola major and minor, also have much shorter branch lengths than you do when you distinguish all of the other species that are part of the genus. This shows you the current tree derived from, that includes the outbreak viruses from 2022 and continuing on to current day. Clearly delineating the clade 1 viruses here that are more highly pathogenic, and then the clade 2 viruses. The viruses from the 2003 outbreak are here. Viruses, the dates that are before 1970 would be viruses from primates or other rodent species usually. The primate infections are themselves zoonotic. More than likely a rodent species is the host species, but that still hasn't been definitively determined. Now, when you look at the dates here, whereas maybe there's a little bit of phylogenetic signal according to date, for the most part, they're all clustered together. So you see 1958, 1968, not too far apart from 2003. Here we have date 1978, 2006, 2023 up here. And so there's not a strong temporal signature to the subspecies clades of these monkeypox viruses. Also, another defining feature is these long branches here between clades. So whereas, the first virus that was isolated that we know about was in 1958, all viruses subsequent to that belong to these very distinct clades. And even in the current outbreak that we see here, we see that divided by long branches that are striking because of how closely compact these other viruses are here. More than likely, this is due to the fact that humans are a zoonotic host. And so there is replication, and perhaps the bulk of the divergence that we see may be occurring in the natural host. Okay, and so we have clade 1, clade 2. We have distinct phenotypes for each one of those clades that play out in what we see now. Now, just to touch a little bit on the mechanisms of variation that we're looking at, orthopox viruses tend to very much be dependent upon gene fragmentation and removal as the basis for the differentiation between the different species. And you see here in red, these are genes that are not present at all in the species that are shown here, whereas at the top, cowpox virus, which tends to have every gene every other species has, is intact. And then dark green, those are genes that are fragmented. Yellow are fragmented even more. And so you can look and see, whereas variola virus has the most genes that have been removed or fragmented, and cowpox the least, monkeypox is in here in an intermediate position. So gene fragmentation and loss is very much a driving factor in the evolution of these viruses. And you can compare and contrast the genes that are present in one virus species or another. Variola has 8 unique genes in comparison to monkeypox. Monkeypox virus has 20 unique genes in comparison to variola. And you can compare the genes between the two distinct clades, 1 and 2 of monkeypox virus. What you see here, there are really only 3 unique genes in clade 1, whereas clade 2 doesn't have any unique genes. And you can try to focus on these genes and do the laboratory experiment to explain the differences in pathogenicity, but in date, there is not one key gene that can define the difference in pathogenicity between the 2 clades of viruses here. And in fact, we don't really know what makes variola virus that much more of a pathogenic human-centric virus than something like monkeypox. So it's probably a combination of genes when you're talking about what defines the pathogenic properties of these viruses. But we do know that within the 2022 clade, clade 2 here, as you see here, actually clade 2b, that there is a distinct evolutionary signature that you can pick up based upon selection of the virus to overcome the ApoBec3 system in the host. And so you see the increase in mutations very distinct in these lineages after they have been replicating in the human host. So that is picked up easily when you look at the impact of selection pressure on these different viruses. But what we don't see, what we can't explain really yet, what we don't see in clade 2 is any distinct signature of pathogenic differences between those viruses. So to date, we only have differences between the clade 1 and 2 viruses that we can actually study experimentally. They lead us to assume clade 2A is more or less pathogenic than 2B. So evolution of monkeypox virus, it's very host dependent with long periods of variation, probably in an unknown reservoir host with periodic zoonoses into alternative hosts, including humans. The mechanisms of variation certainly include SNPs and small indels and ApoBec3-driven mutagenesis, but the bulk of it is due to fragmentation of genes throughout the whole genus.
And so we'll take questions from the audience first. Please.
I'm Christian Smoltek. I work at the JCI and BV-BRC. And my question is for Elliot. I'm very interested in the concept of evolution by loss and not by gain. And so you're stating that the various viruses, they lost genes or proteins. And so do you think you said the cowpox one has the most complete set. So you think that one is the most ancestral one in a way?
So one would certainly think so. And obviously, the progenitor to all orthopox viruses, based upon what we see here, would have had that complete set of genes. And then by progressively losing their genes, then we see this distinct species we have here. Cowpox, though, is kind of a mess right now. It's represented as a single species, but that definitely is polyphyletic with respect to the genus. And so we really need to revisit cowpox virus because there are probably at least 3 and maybe 4 distinct species that are at different points in the orthopox virus tree that we have to reconcile. But they all have this sort of superset of genes.
Okay, that's very interesting to me. Maybe because I did research on this issue before, so maybe I could actually analyze them and try to reconstruct the ancestral genome based on the proteins.
I should emphasize that whereas gene loss is a driving factor in the orthopoxvirus genus, gene gain, that is acquisition of the genes from the host, is the major factor in the evolution of the family.
Yes, I know that there's a lot of, like, for example, like BCL2 proteins in there, and they duplicated a lot, and those are derived from the host. Yeah, it's very interesting. Yeah, thanks.
Please.
Just a very minor question, really. The variola virus may have been cultured. Could it have lost genes during cell culture? I mean, herpes viruses shed them like mad.
So all of these, if they were cultured, it was only 1 or 2 passages, so it's very limited.
Any other questions from the audience? Richard.
So what drives gene loss evolutionarily? Like, what are the selective pressures? Because you would typically think that they would want to gain new functions, but
So when you look at the genes that tend to vary the most within pox viruses, the genes that are lost or gained in the family are generally genes that in some way interact with the host. So immunomodulatory genes, for example. And so it looks like, and pox viruses are very species specific. So throughout millennia, they've co-speciated with their host. And so during that process of co-speciation, the pox viruses are probably fine-tuning the gene sets that they carry to be specific for what they need to best grow in that particular host. So whatever immunomodulatory gene best enables the virus to escape the immune system, that's the one that they pick up. And then they're great at getting rid of extraneous information. So they'll lose genes very quickly, I think, if they don't have selection pressure to maintain particular coding sequences.
Yeah, I mean, SARS-CoV-2's lost a gene or two as well after it got into humans, presumably because they're pointless if you're not a bat, So.
Please.
I'm sorry. Okay, there we go. Okay, I was going to ask about the avian influenza, and I was wondering if in the wild birds, if there's host associations with the viral clades, or if it's just kind of a blur when they're moving through the wild bird populations.
It's a little bit of a blur. It seems like there's some of the holoarctic species. They may be diverging, especially with the group that came to from 2.3.4. That's gone. That's now globally distributed. It looked like there was a little bit of evidence there that it was separating, forming a different group, but that hasn't persisted. So there's lots of interspecies transmission. But we make the assumption in all of our phylogenetic analysis that species doesn't really matter, that it goes between any duck species and any goose species or any gull species, and it doesn't actually drive any sort of diversification. And that's probably wrong, but it's probably not entirely wrong, I should say, but it's not entirely right either.
Yeah, Richard Neer here. I had a question for the segmented virus, like the three of you that are on this side. So for the rotavirus, the rotaviruses, you had classification for each of the segments, right? And this is something that in flu doesn't really exist to the same level. But I think for the H5s, it's sort of becoming more and more of a thing. There's lots of reassortants, right? And people don't quite know how to get on top of that. Do you guys have sort of recommendations or ideas how to tackle that? Like how to go to like genotype constellations, right? Sort of to do that in a consistent and communicable way.
You want to answer that?
Sure. For influenza and swine, we use the same sort of framework. So we're relying upon evolutionary history, and we can track particular internal gene segments across evolutionary history and use names that way and label it with the numbers and letters in a similar manner. You can also use other types of approaches to look at reassortment as well that don't rely on topological incongruence. And identify when particular genome constellations are persisting, increasing or decreasing in frequency as well. So yeah, but evolutionary history.
Yeah, I guess from the rotavirus perspective, for Group A rotavirus, we've found it very important to be able to have at least some mechanism of just describing genome constellations, because we're seeing more and more that particular emergence of strains having unique backbones that we didn't previously see, particularly looking in the vaccine era and this new pressure that might be creating a different ecological niche and different changes of virus dynamics. Being able to describe the different constellations that we're seeing in a manner that is very easy for people to understand which genes are being swapped in and out is beneficial for a field, but how to do it in a consistent way is very, very tricky, and how to visualize that, apart from having 11 trees or tanglegrams, it's not nice. Like, nobody wants to see that.
And the non-segmented guy weigh in.
That's good.
All right. So I have been participating in a bunch of discussions for bunioviruses, which are like the nice segmented viruses, because you typically have only 3 segments and not this nonsense here. So for those viruses, there's at least a discussion going on how to deal with this, and it sounds at least relatively straightforward. So as I mentioned before, all these RNA viruses, in a negative sense, they are classified based on the RDOP palm domain. And so if you make a decision that this is the large segment in bunioviruses typically. So if you make a decision that the large segment is species defining, and you know that the RDRP must, in negative sense, RNA viruses interact with the NP protein, so it must in some way co-evolve with those two. Then the only segment that really gets swapped in and out is the middle segment, which is typically the glycoprotein. So you could make an analogy to the old Haeckel definition of species and make a sex equivalency, that you put several viruses with different names under one species umbrella, and they all change only in the up segment. And then, of course, you could very easily label them by N1, M1, L1, and then N1, M2, L1, and so on. And so that seems to be possible for the simple virus.
So this is when I first got into this field, that was my job. And we would print out all the trees, and we would take colored marker and make a little symbol. I'm older than I look, right? And we would put this across all the trees. I don't want to do that again. That certainly. But we would map all the internal genes against the HA, right? And then we would first the HA, and then we would do it independently for the NA, and hopefully the HA and NA matched up, right? Because especially for H5, right? The relative amounts of reassortment is different for different subtypes. H5 does not reassort that much for an avian influenza. Low path avian influenza in wild birds, you can treat each phylogenetic tree as an independent phylogenetic history, because they are so different. So you could use that as a tool for maybe an ecological study, but you can't actually use that it's almost not useful or practical to trace genotypes for low path avian influenza. For high path avian influenza, we follow the HA. When we looked at, and I think it's the same thing that they do for swine influenza, right? They'll follow the HA first and then map the internal ones against them. Visualizing them, people often will have a tree and then 8 separate columns that have different colors to show that what lineage they might be associated with. So you could rapidly look at a single branch and see that genotype is the same as its neighbors or not, all against the HA though.
And I'd say the last approach that we're probably using, in addition to what Justin just mentioned, is phylogenetic networks that consider the evolution of all genes together. It becomes a tricky computational problem though. And given the scale of data that we're able to get with viruses, there's a bottleneck with phylogenetic network inference. So I'd say that might be a future goal there. Yeah.
Please.
Question from audience, anonymous user. Looks like directed for Elliot. Any information on whether the long branching of monkeypox tree is linked to the intragenic recombination of genes?
So poxviruses certainly recombine. They love to recombine, but generally they'll only recombine. Are you?
Microphone.
Your microphone is on. I'm sorry. Yeah, poxviruses certainly recombine, and they can do it a lot, but it's generally going to be within closely related viruses within a species. And sometimes then it can be difficult to detect because you're dealing with sequences that have only a very few variants within them. I don't think we know enough about what's happening in those long branches to be able to decide whether or not there's recombination. There's just not enough sequences at those points to know whether recombination is driving that at all. And to date, I don't know of any recombination signatures that really explain many of the branches coming off the monkeypox virus tree.
Any other questions? So I have a question. So one of the things that's kind of curious to me when I look at the current state of naming schemes, so we'll just call them naming schemes, is that there is a focus on specific parts of the genome as sort of signals that are most important for consumption on some level. And it's, in fact, as I recall, the rotavirus scheme, that it was actually very much like flu initially. They were really interested in 2 segments and said, oh, we might as well just do them all because it makes for a nice evolutionary biology study when you do that. And then the adenovirus study is really focused on 3 genes. And so a type really becomes monitoring these 3 genes and the alleles of those 3 genes. So is this a model that I'm kind of priming Elliot's section, but is this a model that translates into the future? Or the other models, one of which was just mentioned, are they going to be the things that give the community what it needs, or the means to give the community what it needs going? And what's important about the genome? Let's look at that part versus something that's more holistic and takes into account, one, takes into account the fact there are many more nucleotide sequences available to build the models upon, but 2, takes into account evolutionary history and is more than just marking things A, B, C, or D. Any thoughts from the panel on that?
Well, for adenoviruses, to keep the old timers happy, to X what we're trying to do, but it still makes sense because the 3 markers span the genome. But there are indications that an internal region, the E3 region, is more important. That modulates the host response. So there are some arguing that that's probably more important than just look at major capsids.
Yeah, and likely critical information for a set of stakeholders.
I think I was just going to just say that it depends. You want to essentially place a virus on a map, and how many dimensions do you require to describe the placement of that virus on your map? And so if it's 2D space, 3D space, in biological complex systems, you may need 3 dimensions to describe explicitly where something is. But you can keep on adding more dimensions, and you may have better resolution on that map. It would depend on what your specific question is.
Yeah, and I think that's the key.
Yeah.
One could imagine building a underpinning that's n-dimensional, if you will.
Yes.
That can be compressed to lesser dimensions for the consumption by specific stakeholders, right?
Yep. And everyone knows how to, well, I'm like Justin, older than I look, but I remember opening a road atlas and saying, look at A5 on a map, and then going into A5 and finding a street address. And essentially that's what we're hoping to do with a virus. You could go A5, and then you're on exactly where you want to be on your map in virus landscape, genetic diversity. Yep. Thanks, Faisal.
Please.
I was just going to say, I think one of the reasons why you might pick one region over another is utility. So, for example, in enteroviruses, the capsid gene, or particularly VP1 sequence, is actually what correlates with serotype specificity and obviously the identity of individual virus types. Whereas the non-structural genes are all recombinant. So in fact, it's pointless doing a tree with the whole genome because they recombine so much. So I think, there is a utility aspect to that for some viruses, at least, which actually governs where you would look.
Yeah, agreed. And also the focus, you're moving away from a lot of noise to something that's very focused to information for
That's right,
a certain
yeah.
stakeholder group,
Yeah, sure.
for sure.
Yeah.
Please.
Just Betty Korber, just another comment along those lines. For coronaviruses, people typically use RDRP, that polymerase. But if you try to align spike, I don't think homology is very clear at all. And you can't actually know that you've got a homologous region. So that's the most conserved part. It makes a lot of sense to use that as sort of your phylogenetic mapping because you can be confident of the alignment.
Yeah, yeah. Last question. We have 1 minute left.
Thank
Use
you.
it wisely.
Derek Smith. While we have all of you from different families here, different.
A little ICTV humor, I see.
I wonder whether or not in any of your domains it's important whether or not there are virus mixtures and that there is transmission of virus mixtures. There was a while for one of the dengue viruses, I forget which one, but it was a series of isolates in Hong Kong where there was a second genome that was going along that had a functional stop codon halfway down. And so obviously there was some complementation going on and what needed to be written there for that virus was about the mixture, and the mixture wasn't just an accident and it wasn't just a quasi-species, it was an actual functional mixture. Is that anything that comes up for any of you to have to deal with?
No, it should come up. It's not really coming up. I mean, this is a little bit similar to the whole genome formula of multipartite viruses, right? Where you have these different sets infecting you and it doesn't really do anything. And then another set infects you and then they work together, and you meaning being a plant or a fungus in that moment. So I think this is an important thing, but I don't think that in most virus families, people have been thinking about defective interfering particles being actually functional particles. I think the term is leading people astray, not to think about them. They should. I don't know about you guys.
If I took a guess, I think for influenza, it might play a role within host evolution, but I haven't heard of it being an important thing for transmission across. In fact, I haven't really considered it before you mentioning it.